Good afternoon, everyone. Welcome to this late in the afternoon seminar. It's great to see so many people here and a number of people online, 18 of you. Welcome. So this afternoon, firstly, I'd like to acknowledge on behalf of TRI, the traditional custodians of the land we are on today, the Turrbal and Jagera peoples. We recognise their continuing connection with land, waters and community and pay our deep respects to them and their cultures and elders past and present. And I'd also like to welcome any First Nations people who are here today. So this afternoon, well, the last few months we've heard a lot about Sanofi. Some people have probably not heard much about Sanofi, but we know all about Sanofi now and we're going to learn more this afternoon. So the Translational Science Hub is a collaboration between Sanofi, an international biopharmaceutical company, and the University of Queensland and Griffith University in collaboration with the Queensland Government. And importantly, over the next few years, Sanofi and the hub will be based within this building in TRI and ultimately in the new building behind TRI in TM at TRI. So I'd like to take this opportunity to welcome again Iris DePaz, who's the country medical head. And also welcome to many of you for the first time, Dr Saranya Sridhar. He's the head of translational medicine at Sanofi Vaccines and the global head of the COVID-19 and mRNA vaccines clinical franchise. He's led the clinical development program to license Sanofi's COVID-19 vaccine. By way of background, as head of translational medicine, his team focuses on translational clinical research and early development to optimize mRNA vaccines. Started his career in India, continued his research career in the UK, and now in the US, and now is based in Paris. So we'd like to welcome both Iris and Saranya. Saranya is going to lead off with a discussion now, and we will have time for Q&A. And if you've got questions and you're online, please put your questions in the Q&A box.
Good afternoon, everyone. Thank you very much. And lovely to be here again. We were here in November or December last year, and it's just wonderful to be back. And thank you, Scott. We now know we're going to be based here at the TRI, and we look forward to that over the next few years. I think Scott's done this, and I'm just going to get into, and just on behalf of Sanofi as well, I'd just like to reiterate our own acknowledgement of the traditional custodians of the land. So Sanofi is a large global company. We're present in about 100-odd countries, have 100,000 employees, and we have manufacturing sites across the world. But I think most importantly today, we're very happy that of the 20 R&D sites, one of them is going to be based here at the TRI. So looking forward to that. We have a very old and sort of tradition in vaccinology at Sanofi. We have a very large team that works on vaccines globally. As you can see, roughly about 15,000 employees. And I think what's important for us is we do invest a huge amount of money in R&D in vaccines. And that sort of translates really into a large number of doses that we supply to try and combat some of the public health needs that we have across the world. But I think what I wanted to spend a little bit of time on what we're going to talk about a little bit today later on through the question and answer, but also through the networking session, is what we're doing in our R&D, specifically in terms of mRNA vaccines. And so Sanofi we've had a sort of good experience in multiple different platforms and technologies. And one of the things we're trying to show you on this slide is the fact that we are reasonably agnostic to the kind of platform we use for the right kind of pathogen or the right antigen and the right indication. But very clearly, after the COVID-19 pandemic, we recognized the need for developing mRNA vaccines. It's pretty obvious during the pandemic, and that's really what we've now been accelerating over the last few years. And there's many different advantages of mRNA vaccines, and I'm not going to get into that. That's perhaps a chat for another day. But what I do want to show you is some sense of what our R&D vaccine pipeline looks like. And what you can see here is we've got a lot of different products that are in different stages of vaccine development. A lot of focus, as you can see here, on respiratory pathogens. We've got RSV, we've got flu, we've got pneumococcus. Very proud that we've got the first monoclonal antibodies coming out for infants for RSV, nirsevimab, which is really fantastic. And we've just recently licensed our COVID-19 product in Europe as well. But what I think I'll be interested in spending some time here is the fact that if you look on this slide and you look against all the things that have a sort of purple line against it, chlamydia, acne, RSV, flu, and we also have an influenza monovalent vaccine, a large amount of our portfolio is now going to be based on the mRNA platform technology. And what we've committed to over the next few years is to have about 60% of our vaccine portfolio really focused on using the mRNA platform technology. And what that meant for us was really a huge amount of investment in the mRNA platform technology. And we'll talk to you a little bit about that. So when we came to the problem about mRNA, the first thing we recognized is there's perhaps a long way to go. I think the technology, even though it's been worked on for the last 20 odd years, is to some extent still in its infancy when it comes to actual use. And so we decided that we needed to really accelerate our own efforts on trying to improve the mRNA platform that's in our hands. And we set up, therefore, this mRNA Center of Excellence. We started in 2021 and really turbocharged the ability to set up this entire Center of Excellence. And I'll talk a little bit more about that over the next few slides. But what that really meant was when we decided to have that idea in July 21, we wanted to be able to execute that in a year's time. And over the last year or so, we've had about 400 different employees now part of the mRNA Center of Excellence across 2 different sites in the US as well as in France. And we've committed a substantial amount of investment to this platform. We're looking at about investment, about 400 million euros annually every year on trying to develop mRNA vaccines. And we acquired a company called Translate Bio, which was based in Boston, a US company. And that really, again, was a really important sort of acquisition for us, because what it allowed us to do was internalize the efforts we wanted to make in improving the technology. So this is where we are today. It's both in France and the US. So if you ever turn up in Waltham, which is near Boston, please look us up. We'd be happy to show you around. And we're also based in Lyon, which is our major R&D site for Sanofi vaccines. And so we're based there as well. So both these places are which houses about 400 of our staff who are focused on mRNA vaccines. And what are we trying to do? So what we decided to do was really empower the Center of Excellence to go all the way from the very start, from the design of the mRNA itself, all the way as to the clinic. So you can see here, we go right across the board. And the idea was that as you get towards the end of this line, we'll go through a little bit about these intermediate steps. Really, the idea was how fast can we get into the clinic, and how does that then translate into actually coming up with multiple programs and multiple products. So just focusing on the RNA science itself, we have an entire team that's focused on really trying to improve the mRNA sequence. A lot of what we do is this idea around codon optimization. So we do spend a lot of effort trying to ask pretty simple questions. How do we improve protein expression? How do we improve stability? How do we improve purity? Can we improve thermostability as well? And how do we, most importantly, lower the reactogenicity of some of these vaccines? And we think understanding how to optimize that sequence is going to play a very important role there. And just on the bottom slide, you can see we've done many different attempts at different types of codon optimization. And of course, I think there's a lot more to do here. Nanocarriers. So of course, lipid nanoparticles have now become sort of lexicon that everyone can speak to, but we've spent a lot of time and are spending a lot of time trying to improve the delivery mechanism for the mRNA. So we have a very large library of lipids, and we're constantly trying to enhance those. And some of the things we're looking at is not just sort of different types of lipid families. We're trying to also try to understand how to improve the delivery of these lipid nanoparticles as well. So different routes of administration is something we're also trying to do. And again, you can just see here, we've got a library of about 700 lipids, and we're constantly expanding on that. And the idea is, how fast can we understand and design lipids much better for the future? So one of the questions we're really interested in asking as part of the TSH is, not only can we understand the mechanism of action for some of these lipids, but how do we back translate that so you can then improve the design of future lipid families and other delivery mechanisms? A lot of what we do, as you can see with the fact that we're doing a lot of different codon optimizations, a lot of lipid work, but many other things we're looking at is really sort of based on the foundation of really looking at automation and looking at data sciences. And that's really important for us as we try and generate more and more data over the next few years. And this is just a really interesting slide. This was done by some of our researchers, and they said that if you just take the hemagglutinin sequence of flu virus, you can have roughly about 10 to the power 273 distinct sequences to try and improve some performance parameter of that particular sequence. So there's no way we're going to try and make that number of sequence, but what it shows you is the scale of work we might have to do to try and improve the sort of performance of our vaccines. If you combine that with all the different lipid families we're going for, it just generates a huge amount of data. A question for us is, there's no way we can do this in a sort of, if I may say so, 1900s sort of empirical vaccinology model. I think we've got to find a better model of trying to improve these mRNA vaccines. So we're really interested in looking at artificial intelligence and machine learning that allows us to sort of really understand what are the preclinical parameters that might predict some of the performance outputs that we're looking at from our vaccine. And the idea behind that is, if you can find these preclinical translational parameters, then your down selection pathway to get into the clinic becomes so much faster. That's one part of it. The second part of it is it allows you to really start designing these vaccines a lot better. That's something we're really looking forward to. So where are we on our journey, and where does the hub then fit in? So we believe we made a lot of progress towards a next generation mRNA platform for us at Sanofi. You can see some of the things we've done over the last year. This is literally a year of work here, and we've got a lot more to do. We're focused on a number of different lipids that we can look at, that we can identify. We've started about 7 different clinical trials last year, just on mRNA vaccines. We plan to do another about 7 or 8 this year, and so on and so forth. And the idea there is to really try and go into the clinic as rapidly as possible to try and generate the data that we need to try and improve the platform. A big question for us, and I think this is something we really look forward to at the TSH, is really understanding this concept of reactogenicity of mRNA vaccines. So we really think there's a need to really not only understand the mechanism of action, understand some of the biomarkers, but then go back and again redesign these vaccines to try and lower some of the reactogenicity, the side effects we see after mRNA vaccination. And of course, I'm sure all of you've been following the pandemic. A huge problem for us when it came to actual deployment of the COVID mRNA vaccines was the issue of the cold chain. And so one of the issues we have started looking at very significantly is, how do we get our mRNA vaccines to become more thermostable so you can actually use at 2 to 8 degrees cold chain storage to try and deploy these vaccines when they get into the field. So big research areas for us over the next few years. So this just gives you, I hope, an overview of where we're trying to go in our endeavors. And I'm going to spend a little bit more time, perhaps, getting a little bit more specific of what we're going to try and do while we're here at the TRI of the next 5 years. So the Translational Science Hub, as Scott said, was a real partnership between a number of different organizations, between Sanofi, between the universities, between the government as well. And when we looked at what we wanted to do through this Translational Science Hub, we really landed on a couple of big projects that we thought we want to take on, at least in the short term, as we try and then establish it and make it more sustainable. So the first thing we really want to do was ask, how do we improve mRNA vaccines as a platform? So what we believe we are, and the situation we are in today, is we've got an mRNA platform that works for pandemics, outbreaks, that probably does absolutely fine. Where we need to get to is a platform that we can start using for more endemic seasonal use. And the question is, how do we get there? And in that process, we really want to get to a third generation mRNA, which probably is hopefully Translational Science Hub version 2.0 over the next few years, where we really can try and identify and target these mRNA vaccines or mRNA therapeutics to very specific cell types and particular parts of the body. So how are we going to do that? So we've designed a kind of umbrella protocol, which is a clinical trial protocol, which is very common in oncology and other areas of research. For vaccines, what it simply means is that you have one clinical trial protocol. Then as you try and iterate and bring up new interventions or new ideas, you can run it through the same clinical trial protocol. And what we're committing to do over the next few years is have roughly about 2 trials every year to try and improve this platform under this umbrella protocol. This is a very bad figure, but this is roughly what it looks like, is this idea that you decide what your inputs are, you try and change your vaccine in specific ways. And because you've got this umbrella design where you can put in different interventions, you can have interventions that drop out, you can have interventions that stay in, but you potentially have a control group that stays the same. You then begin to understand and generate the data very rapidly in terms of some of these outputs. But the fundamental purpose of doing this is that you try and generate this clinical data as quickly as you can. So you start really understanding some of the preclinical parameters that actually translate into performance in people. And I think that's really the big question we've still got outstanding at the moment. So that's one part of it. I think we're really also trying to understand through this umbrella protocol, what is the mechanism of action of these mRNA vaccines? What's the balance between the innate and adaptive immune response? What is the balance between the amount that you need for immunogenicity versus reactogenicity? And most importantly, if you understand that, perhaps how do you begin to start modulating that in the design of your vaccines? But to do that, we really need to understand the immune system a little bit better. And a large focus for us over the next few years, and I think part of a big reason for us to be based here at the TRI was really to be able to link in to some of the immunological expertise and the clinical expertise and research expertise at these institutes, as well as, of course, in some of the universities we're going to be partnering with. So you can just see here, and I'll show you another slide of the kind of things we're going to do. But the question we're really asking ourselves is, how do you start looking at data quickly enough so you actually predict what the vaccine is going to do in the real world? And that's a real challenge for us. We do phase 1 studies, we understand it. In a mouse experiment, you can understand that. You can do in vitro screens. But the real question is, when you start deploying these vaccines in hundreds of millions of people, how do you actually try and predict what that response is going to be? And the question for us is, how do we generate that data, that translatability sooner rather than later? This just gives you a very quick sense. These are all the studies we've started last year, a few that we're starting this year. I don't want to spend too long, but I just want to give you a sense that of all the different immunological readouts we're trying to get, to really try and understand to a very in-depth level what the immunological response looks like post-vaccination. So of course, you can see here a lot of different cool assays, but the purpose of these very cool assays is really to try and tease out what it actually means. And again, I come back to this feedback loop, which is really, really important to us. So one of the biggest questions we're asking ourselves is, how do you translate? What are the preclinical translatability parameters to the actual clinical performance? So the question for us is, we have these screens, we are doing a lot of expression screens, we do in vitro screens, we do animal studies. And then when we get to the clinic, the question is, which of these different things we've done to down select to get into the clinic actually translate into clinical performance? And then we back translate that to try and design vaccines a lot better. And while we're based here at the TRI, one of the things we're really looking for is, how do we start doing these trials so that we can get data faster? And so we do small trials, we get the data quickly, we try and get the data that's in depth, and then really try and back translate that and go back faster in terms of design. The second big objective we have with the Translational Science Hub is a problem that I know most of you are aware of, which is chlamydia. It's a huge WHO priority in terms of sexual health. It's the most frequently reported sexually transmitted infection in Australia. And it's something that we believe is a real challenge for us when it comes to developing a vaccine. So what we're really looking at through the TSH is actually a full clinical development of the chlamydia vaccine, all the way from phase 1 all the way through licensure and phase 3. And there are some really significant challenges in trying to do this. We really don't understand a good correlate of protection. We really don't understand how to get the right thing to the mucosa. We actually, to a large extent, don't even understand how we're going to look at the endpoints for this particular vaccine. So a large number of things we really have to do as we get towards trying to make a mRNA chlamydia vaccine. And I'm coming to the end of my talk, but I do want to spend some time talking about how we want to work here. And I think the foundation of trying to achieve these two objectives around improving the platform and the chlamydia vaccine is really the foundation is the ecosystem we're going to work in. And so we really wanted to make sure we were embedded within this ecosystem and sort of leverage the strengths that we had here. So a few things I will talk about in terms of what we want to do. The reason we came here is we know that Australia, we've been doing trials in Australia for a long time. The clinical trial capacity here is great. But what we do want to make sure we do is enhance and increase that capacity over the next few years, both in terms of looking at phase 1 clinical trial units, but also other clinical trial units as well, with the idea that we can really sort of develop a more sustainable platform for us in terms of clinical trial operations to run some of these studies over time. We're really interested in doing a lot of laboratory research, and we want to try and build that laboratory capacity here as well. We, of course, can tap into the fantastic core facilities that we do here, which is with all the phenomenal flow cytometry and other techniques, but we also need capacity to do some very boring antibody assays and ELISAs at a GCP and qualified level. And we're going to have to build that capacity over time to kind of sustain the kind of work we want to do. And one of the things we really want to focus on is, and I think this is why we want to have these meetings and we want to encourage all of you to come up and talk to us and try and encourage us. We want to engender long-term collaborations with the universities here, with the researchers here, with the biotech industry here as well. So really finding opportunities to collaborate where there is mutual benefit, there's mutual interest, I think is something we're looking forward to over the next few years as well. My next 2 slides, and that's my last couple of slides, is really pitching to you that we are hiring. So there's a number of, we're going to have about 15 individuals in the Translational Science Hub with a large number of different roles. And I'm just going to share with you a few of these roles and give you some understanding of how those roles fit into our global R&D organization. So when we do our studies or when we do any particular project, we have a sort of core clinical team, and we're hiring for clinical leads, which are sort of clinical trial physicians. We're looking at clinical study managers who run these studies, very interested in biostatisticians, data scientists, and really looking at immunologists as well, particularly people who have a sort of interest in both immunology and epidemiology. And when we build that kind of core team, they all become part of a much larger global project team that is really global in the sense of you have individuals and teams that come from large different parts of the world. That project team is the team that fundamentally drives the development of that particular program or that particular asset. And as you can see here, it consists of many different expertise, from medical to regulatory to industrial to sort of understanding commercial strategy. And the advantage you have when you come and become part of that global R&D is you really get a sense, a very deep understanding of what it takes to really get a vaccine and get it into people's arms. And that's something that's really important, and we believe we offer if you come and sort of collaborate with us. We also are hiring for a number of different operations positions, which is very, very important. We have a very key position as a director of clinical operations that we're hiring for, a project leader for the Translational Science Hub, somebody who manages a large number of the partnerships we hope to build over the next few years, and some of the other positions you can see are data managers, medical writers, etc. So the point of this is the thing on the top right there, which is we are hiring. There will be openings coming out, so please keep a lookout, tell your friends. And not that we want to poach some of the amazing staff here, but we'd still love to collaborate. Let me end by just saying what and how can we collaborate with you? And I think this is going to be very simple, but we really encourage you to come and talk to us. We really want to learn what you're doing. We're very keen to share what we are doing, and really want to encourage that kind of open transparency and communication and exchange of ideas. University collaborations and institutes are going to be part of the Translational Science Hub. It's part of the project plan. It's within the agreement that we have. And so it's important that we look towards trying to establish these collaborations. And that ranges across the board, all the way from doing very early research, all the way through assay development, but even looking at sort of statistical methods, real world evidence. There's a range of stuff we can do. Iris DePaz, who's our country lead. Iris, put your hand up and say hello. So if she's here in the green dress, but please talk to her, because we're really looking to try and develop industry placements. We're trying to develop educational courses, potential fellowships. If you're doing a sabbatical, come and work for us or work with us. That'll be fantastic. We're looking at partnerships, joint funding applications as well, all part of the TSH. So I think it's really important, and we want to collaborate more with you. I think what we can offer you, if that's helpful, and if that's of interest to you, is access to a large global R&D organization. And that's not just vaccines, that's beyond vaccines. We have huge interest in specialty care, in immuno-oncology. We have a large amount of interest in rare diseases. So a huge amount, and immunology is a huge focus for us as well. We also give you the opportunity potentially to operationalize some of your ideas. And I hope that's something that you do take us up on. So if you have an idea, please come and talk to us. We'd be happy to see if through the TSH we can make that actually come true. And the last thing we say is, while we're going to be based here, we are going to have a local translational medicine unit that can actually run clinical studies out of it. So the opportunity to actually do these clinical studies as part of the TSH is, I think, is very important. And lastly, I'm sure you're aware that as any large developer, we do have a department that is really focused and looking at opportunities, looking for opportunities in terms of licensing arrangements, looking at opportunity in terms of partnerships, but both for products as well as for technologies. So with that, thanks for your attention. But my main message, if you don't take anything else out of this, is please come and talk to us. Thank you very much.
Okay, so Saranya's happy to field any questions. Where do you see the chlamydia research going in terms of clinical trials over the next couple of years?
Okay, good. So, the challenge for us in chlamydia is, I think we don't understand the bug as well as we think we do, which is the first thing. And I think the immunology of that is tough. One of the first things we're going to try and do is start an epidemiology study, hopefully later this year, early next year. And the sort of idea around the epidemiology study is not just to understand the burden of disease, but really to understand some of the natural history of disease in context of the immunology. So what are some of the correlates of risks, some of the correlates of protection as you transition from infection through to more chronic diseases? And that really leads us into a phase 1/2 study early next year, where we've got a candidate vaccine that we're going to put into the clinic. And the question for us scientifically is, when you go into our phase 1/2 and you measure some stuff in your blood, whether it's antibodies or T cells, the question for us is we don't know what goodness means. So how are we going to find out whether it's actually the right thing that we're measuring or not? And I think that for us over the next few years is the big scientific challenge as we go into phase 1/2. Assuming we get past that, we are going to be looking at a phase 2b proof of concept study and then a sort of global phase 3 efficacy study as well.
So we have a question online. We have a question from DU here. While we're waiting for that question now, Ben Baker from Vaxxas, obviously biased, but alternative delivery systems, are they on the radar? So you've got a potential collaborator already.
Yeah, absolutely, they are. And actually, we're very interested in alternative delivery mechanisms, but also routes of delivery. I think we've got a, Translate Bio, which was the company we acquired, was actually looking at intranasal pulmonary delivery of mRNA for cystic fibrosis. So we're very interested in sort of expanding on that and really understanding whether we can give vaccines through many different pathways, including patches. So hopefully, I know we've already been in conversations with Vaxxas for some time, so we hope we can take that collaboration much further through the TSH.
Thank you. This is Dee from Fraser Institute. And the idea of umbrella protocol is really fascinating. I just have 2 questions around those ideas. The first question, when you do clinical study to try to optimize the umbrella protocol, do you have any kind of design about the size of the cohort, consider heterogeneity in individuals? That's my first question. The second question is, once you have optimized the protocol, do you think from the regulation perspective, there was a most likely technology platform-based approval rather than individual clinical trial, not ready now, but in a few years' time from the regulation perspective?
Yeah, it's a great question because it's, I think take a step back. I mean, platform trials and these umbrella trials have been going on for some time. This is not particularly new, particularly for oncology. One of the most famous ones more recently done was the recovery trial in the UK for COVID, where they tested many different interventions over time and was able to really show in real time what worked and what didn't work. The challenge for us is what we're really trying to do is take that a little bit differently, but doing it in healthy individuals in early phase 1 studies, which is where the challenge lies. So the answer to your question around the sample size and the heterogeneity is a really important one. And one of the things we're beginning to slowly learn is when you look at subjective measurements like reactogenicity or side effects, you actually need to be very clear in your inclusion-exclusion criteria to sort of try and limit that heterogeneity. That becomes obviously equally important when you look at immunogenicity, but there I think you have a little bit more leeway because you can really restrict previous infections, you can look at previous vaccinations, so that becomes a bit clearer. So the answer to your question, I'm taking a long route around, is you can do small studies and you don't have to predefine exactly what number you're looking at, but we rarely would do a sample size estimations, usually down to feasibility and a phase 1, so about 30 to 50 people at a time. So pretty small studies. Right? And I think the second question around regulation is really important and interesting. I mean, I think there is more and more innovation from regulators to try and encourage us to take very innovative approaches to clinical development, full stop. And I think if you go and speak to the regulators, you'll find that they're actually looking for ideas to come and give them an opportunity to use that as they move forward to either accelerate clinical development of products. So I'm not sure where the platform trial will land, but I can tell you that when we went to the regulators and we did a very similar thing for our COVID-19 program for now phase 3, the regulators were actually pretty open to the idea of using a platform trial. We had two different products that we tested at the same time, and they were very open to doing that for COVID. Whether that stays on for the future and for future indications, I don't know, but my guess is going to be they're going to be very, very open to this.
So we'll bring the mic to you, Michael. We've got another online question. You've touched on oncoimmunology, rRNA vaccine for common cancers also on Sanofi's radar. And that's from Josna.
Yeah, absolutely. We have a huge interest in particularly immuno-oncology, and we're really looking at mRNA as a technology to sort of support our immuno-oncology therapeutic area. I think what we're going to try and do through the Translational Science Hub in the first year or two is really provide that proof of concept that this is a platform we understand and have a sort of good handle on, and really then use it as we go forward across many therapeutic indications, not just restricted to vaccines or cancers.
Michael Nissen.
On? Yeah, it's Michael Nissen from QASIS at RBH. Thank you. I've just got a speculative question, and it relates to the interplay between the nanoparticle and your mRNA sequence. The nanoparticle has received some negative attention in terms of increasing adverse events, but could the nanoparticle, and are you looking at using it as a non-specific immune stimulator in an interplay with your mRNA? Is that technically feasible or not?
Yeah, it's a great question. I think, I just get excited because I think it's going to be a fun time in the wrong way to really try and understand and explore this over the next few years. I mean, I think we have a pretty good idea that I think the lipid nanoparticle clearly has an adjuvant effect. I think that's reasonably clear, and we're clearly seeing that. And there's sort of some increasing data coming out of, at least from Moderna, some of Moderna studies that shows that if you look at some of their COVID data, you had pretty high reactogenicity, but in fact, if you look at their RSV candidate, their reactogenicity is a little bit lower. So there is some suggestion that the interplay between the lipid and the sequence might actually play an important role in trying to sort of determine that balance between immunogenicity and reactogenicity. So I think we're going to have to find a way to understand that a bit more carefully. What we also know from some of the preliminary work we've done is clearly the type of lipid we use also clearly determines the kind of reactogenicity profile we begin to see. So again, the question for us is, are all the lipids that are non-reactogenic also non-immunogenic? Are all the ones that are very reactogenic also highly immunogenic? Or is there there's a sort of Goldilocks zone somewhere in the middle? I think that's going to be the challenge for us to find.
So I just wanted to ask you a little bit more about the Moderna RSV result. From a point of view of the strategy of a large pharma, do you go to find a second but better RSV vaccine now, or do you look for one of the other many infections we're looking for better vaccinations?
It's an unfair question, but I'll try and answer it. So, I think we're actually very encouraged by Moderna's data, to be perfectly honest. I think for multiple reasons. I think the first reason is Pfizer, GSK, and now Moderna have all come out and shown that we can make a vaccine against RSV, which was not something we thought we would be saying in 2023, 10 years ago. So I think from our perspective, we believe we've sort of made the right choice in moving in that direction. Are we going to make an mRNA vaccine against RSV? The answer is yes. Do we think there's enough space? I think the answer is also yes. And I think the question for us is going to be, what's the difference in the different overall profiles of these candidates? One of the things we are exploring is whether we should think about having a vaccine that's combined with some other respiratory pathogens that are not given the same amount of attention as RSV and flu. And so that might be somewhere where we will probably try and build a vaccine against other pathogens as a combo.
Just a final speculative sort of question. Any suggestion that we may go towards niche vaccinology. So the mRNA particle and the nanoparticle suited towards age, but also sex of the individual.
You know, I love the question because I think we will get there in the end. Whether we get there now or we get through the mRNA, I'm not sure. I think vaccinology over the next 20, 25 years is going to get tailored. And I'll give you two good examples of that. I mean, the first one is our dengue vaccine, which we had at Sanofi. We made the first dengue vaccine, but we recognized that when we looked at the performance of that vaccine, it was actually very different between those who were seropositive and those who were seronegative. So that actually led us to try and go and develop a rapid diagnostic kit. And we actually now use that vaccine, at least in the US, in combination with the rapid diagnostic kit that actually allows you to differentiate between people who are previously infected and not. So I think that this idea of tailored vaccination, that context is obviously existing. And this used to happen a long time ago for BCG, by the way, 100 years ago, so it's not new. The second example is actually what we do today for flu. And I don't know, we already have tailored vaccines as you look at by age. So for example, if you look in young pediatric populations, you often go for a recombinant protein or you go for the standard egg inactivated vaccines. As you go through sort of young adults and sort of middle-aged adults, you sort of use our standard vaccines or adjuvanted vaccines. Then as soon as you go into slightly older adults, you begin to use our high-dose vaccine and adjuvanted vaccine. So you're beginning to see this sort of difference in the kind of products you're trying to develop depending on the population you have. And I think that is just going to get more and more refined as we go along and learn more about these vaccines.
I was interested in your comments about doing studies about mechanisms of action. Tell us about the clinical trials that you envisage that might help that, and what do you need from the scientists here specifically?
So that's very exciting. I get excited every time I talk about it. So I think there are 2 or 3 big questions we really want to understand in terms of mechanism of action. And the first question we've got to understand is we've got to solve the problem of understanding reactogenicity. What are the biomarkers of reactogenicity? And how do we modulate that? So that's an important question. And the question for us is, how are we going to understand that better? So do we do trials in different populations? Do we do trials with a recombinant protein and adjuvant versus an mRNA and understand what that innate response looks like versus adaptive response looks like? That's really important for us. I think the second big question we've got to understand in terms of mechanism of action for mRNA is actually durability. We're really beginning to see that perhaps the durability of some of these vaccines is not clear, and we're not sure exactly how to design these vaccines to provide greater durability. I think understanding that, particularly in terms of the early B-cell responses, the germinal center interactions is also going to be pretty important. And I think the third big question for us is, if we go even further, are there and we talked about this a little bit, Scott. I mean, are there studies you can do where you try and use and combine vaccinology with kind of immune modulators, where you can really understand which pathways these immune modulators target to try and dissect out how your vaccine works. And so to come to your key point, which is how are we going to interact? What do we need from researchers here? I think the first thing is we'd really like to leverage some of the immunological understanding and the technology that exists in the labs today. I think doing that is going to become very important for us. Some of the things we're beginning to do, just very basic in terms of omics versus looking at B-cell responses, are important. So trying to get more and more of that kind of data is going to be important. And the second thing is we've got to try and understand how these vaccines work in different kinds of situations. So if you take individuals who have rheumatoid arthritis, if you take individuals with rheumatoid arthritis who are on anti-IL-6, do you see slightly different responses? And what does that mean for understanding the mechanism of action of these vaccines? So I think those kind of studies are probably well suited to the kind of individuals and the thoughts that we have here, and we hope we can actually do that.
Question up the back.
Here in Australia, we love our koalas, and the koala population is declining. And a couple of reasons why the koalas are dying is they being involved in car accidents, and the second thing is chlamydial infection. So do you know some of your research is going to contribute towards developing vaccines and protecting our koala populations here?
Absolutely. So in fact, when we came out last year, we actually went out to the zoo, the hospital. And in fact, we actually spoke to some of the researchers there who were working on trying to make a chlamydia vaccine for koalas. And the question was, what can we learn from them and how can we collaborate? I'll be reasonably clear. I don't think we're going to make a koala chlamydia vaccine today. That's not in the plan. Whether some of the learnings we get from trying to develop that vaccine for humans is going to translate into a vaccine for koalas, I think the answer to that is yes. And I think we'll have to work with some of the research. And, there's Peter Timms, for example, who's been doing vaccine research here for koalas for 20, 25 years. So, we're going to come and work with them and see how we can collaborate and sort of develop that in a mutual way. So the answer is yes, but also perhaps not exactly.
So a question, do you already have the microfluidic ultrastructure to produce a large scale of nanoparticles, or is that something you're looking at investing in? Maybe you should explain that question, at least for me.
Well, I have to admit, I'm not the industrial affairs guy, so I'm not going to have too much. What I can say is that we do have infrastructure where we are manufacturing for our lipid nanoparticles, and we're building up that at scale pretty quickly. But one of the things we are really interested in doing over time at the TSH and while we're here is working with the DMR that's going to come up in a few years about trying to develop GMP material for some of our phase 1 studies, particularly at small scale, that we can then use locally and run these studies out of here. So we are interested over the long term in terms of trying to build manufacturing capacity, but perhaps not necessarily at very, very large scales.
I have another question here.
So when do you think the TSH is going to be operational? And when are you looking to start hiring people?
Great. You've got to the crux of the question. The entire seminar is just, you got to the main bit. So the answer is we are operational very soon. We've just recently received confirmation that we are going to be based here. So as I can say that we've got the lease, so that's really great. We are hiring very soon, and we'd like to start very soon. So hopefully we're going to get the job adverts out over the next month or so, if not in the next few weeks. So keep a lookout, and then we'll be working here. I think over the next couple of months, what's more important for us is we hope that the team that we have here will start living here, and you'll have an opportunity over time to sort of chat to them and interact with them, and hopefully for them to have an opportunity to come and interact with you.
It's very clear that when we talk to the MCRs as the leadership group within TRI, they're very keen to understand what the various options are for their career progression, academic and non-academic, but also opportunities to combine both, and that's where they go into industry. And ironically, we were just talking about this when the email came through a couple of weeks ago about this opportunity here. Of course, advertising that there are jobs in industry in the building puts me at some risk with some of the group leaders because they don't want to lose their best scientists. But I guess the whole idea here is the collaboration and the partnership with Big Pharma, which we really don't have, or we just have at a distance at this stage.
I mean, I think the intention is, the number of people we're hiring is not that many. The point, I think, is really how do we encourage movement, and not just movement in terms of people on both sides, but also sort of a little bit of knowledge transfer, particularly as it comes through in career development. I think as someone who did a sort of stint in a lab a long time ago, I think if I had the opportunity to have spent some time in industry at that time, I think my outlook, both from an academic perspective, would have also changed. So I think it's important, and we're looking towards trying and setting up those kinds of options of having fellowships or placements or stints where we can have individuals come into Sanofi and then perhaps even go back and sort of undertake those kinds of opportunities.
Okay, well, I think we've probably been through all of the questions we have both online and in person. There is a networking opportunity where, if you're interested in getting more information from Saranya or Iris, not only about the science that you might be able to do in collaboration, opportunities for future employment, please talk to them. That's going to be in the atrium straight after this session, which I'm going to close now. Thank you very much for coming.